All right, here we are, video 3 for population genetics. We have calculated our genotype frequencies from our genotype numbers. It's possible that you would be given the genotype frequencies, so that's kind of step 0. Step 1, our first calculation step, was to calculate allele frequencies, and we did that 2 ways, the long-handed way and the shorter way, straight from genotype frequencies. And now what we want to do is ask, what genotype frequencies do we expect if these gametes are sorting themselves out into zygotes in a random way? Okay, so when alleles are transmitted by meiosis and random fertilization, so when alleles are sorted out into gametes by meiosis, and when those gametes find each other in a random way, like when we picked 2 beads out of our bucket of beads, all the beads were the same, you had no bias, you weren't looking, you had no bias into whether you were going to pick red or white, you just picked whatever you happened to pick. Okay, when that's the way that we make zygotes from gametes, the allele frequencies shouldn't change. If we make fewer zygotes than we had alleles, the numbers might change, but the frequencies should not change. So in the next generation, we think the frequency of A, big A, should still be P, and the frequency of little a should still be Q. Okay, the sort of problem, or the reason that we need to use a little bit of our past knowledge about inheritance, genetics, is that there are more than one way, there's more than 10 ways, or many ways that our, in this case, 200 alleles could sort themselves out into genotypes. We could get any of the combinations pictured here. We could get these combinations. We could get many different combinations and still have 120 big A's and 80 little a's, or 60% big A's and 40% little a's. Okay, so how do we know what we expect? Well, one thing that we could do, don't write all this down. Okay, one thing we could do is if we were actually somehow able to observe the matings, and we knew the genotypes for all the parents, we could do all of those Punnett squares or use our probability rules. We could actually figure out the odds of every possible genotype. But we don't know the matings. We wouldn't want to do it that way. That's really laborious. All we need to do is to use our understanding of mating, generally the probability rules that govern mating, which we spent a lot of time doing last week. Use that to predict our expectations. Okay, yay, the multiplication rule. Just a reminder, this multiplication rule says that the probability of 2 separate things both happening is equal to the product of the independent probabilities of those 2 things. So the likelihood of flipping a coin twice and getting heads both times is 1/2 times 1/2. All right. So random fertilization means that the identity of the first gamete is independent of the identity for the second gamete. So if we're thinking about getting alleles like flipping coins, the likelihood of getting either allele is equal to its frequency, right? If we have, let's say we've got 10 beads in our jar, in our bucket, and 9 of them are red, then we would expect that 9 times out of 10, if we take out a bead and then put it back, we'll get a red one. And 1 time out of 10, we'll get a white one. All right, so the logic is just based on probabilities, based on frequencies. Okay, so we can think about a probability matrix before we just put it like the allele, big A or little a. if we're thinking about a population being in this box instead of just 4 individual offspring, if we think about population genetics, we can think about P and Q being on either sides of our probability matrix. So you're going to get one allele from one gamete in our gene pool. The likelihood that you get the big A is P. The likelihood that you get the little a is Q. So before we looked at one individual on either side, and they had 2 alleles, both present at 50% because they just had 2 alleles. So you have a 50% chance of getting one of their alleles, a 50% chance of getting the other, heads or tails. Now, P and Q might not be equal to 0.5. So we are going to have to use our multiplication rule. All right, and we've got this happening. We're imagining that this is happening in a really large population where individuals are mating randomly, mating without respect to the alleles that they have for the locus that we're interested in. Okay, so in our population, where 2 alleles are coming together, 2 gametes are coming together, the odds that you have one gamete with the big A allele is P. The odds that you get another gamete with the big A is P. So the odds that you get a big A and a big A is P times P, or P squared. Okay. Let's go over here. The likelihood of getting a homozygous recessive individual is the likelihood of getting one recessive allele times the likelihood of getting another recessive allele. That's Q times Q. So we're pulling these 2 alleles out of our big bucket of alleles. The likelihood that we get a little a is equal to the proportion of little a in that bucket. And then our heterozygotes, the likelihood of getting a big A is P. The likelihood of getting a little a is Q. So P times Q represents our expectation for this genotype frequency. Oh, but there's another way that we could get a heterozygote. We could get a little a and then a big A, right? So we've got P times Q again. So we've got 2 PQ. All right, so our expected, given in our example with our numbers, our expected genotype frequencies are here on the right. So our expected G big A is equal to P squared. That's 0.36. 0.6 times 0.6. 6 times 6 is 36, so that's how that decimal math works. P times Q is 24. 4 times 6 is 24. We've got 2 of those, so we get 48%. And Q squared is 0.4 times 0.4, or 0.16. Now you'll notice again, just like before, our genotype frequencies add up to 1. This, I'll come back to that gray part. This is because we have 3 genotypes in our population. Some proportion are homozygous dominant, some proportion are heterozygous, some proportion are homozygous recessive. And if we add up the proportions of all the 3 types, we have 100% of the types. So our genotype frequencies, when added, should equal 1. This doesn't tell us anything about evolution. So this is way back in, I think, video 2, I was telling you that sometimes students do calculate and forget to compare. This is where people get off. So, warning. The fact that P squared plus 2PQ plus Q squared equals 1 is just a mathematical truth. This does not tell you anything about evolution. What we have to do is compare our expectation that we just calculated to our observation from before, the thing you were given. Okay, so way back in video 1, I told you that the Hardy-Weinberg principle, or Hardy-Weinberg equilibrium, is summarized by this equation: P squared, the likelihood of being homozygous dominant, plus 2PQ, the likelihood of being a heterozygote, plus q squared, the likelihood of being a homozygous recessive, equals 1. Okay, we expect our population genotype frequencies to equal 1 whether or not the population is evolving. Okay, so the way that we use this tool is not by adding these things up and saying, oh yes, it equals 1, that tells me something. We use this tool because it summarizes everything else that we just did with the probability matrix, or that Punnett square if you want to call it that. Okay. We're coming towards the end of this third video. After this, you're going to have another assessment, and then I will post a video where I work through a problem from start to finish, and I'll encourage you to work through that problem with me. Okay. So we have our expected genotype frequencies that we just calculated. At the beginning of this series, we were given these genotype frequencies. We observed them, or someone observed them and told us about them. All right, now here's the key. We compare our actual to our expected. And you can see they're the same because I made this example up and I made it work. Right? So if that's the case, if our actual frequencies meet our predicted or our expected frequencies, that tells us that the allele frequencies, the gene that we're looking at, is in Hardy-Weinberg equilibrium. It is not evolving. Okay, this tells us that our population meets the assumptions that we make about a non-evolving population, and we're coming to those, I think, on the next slide. If our actual frequencies are substantially different from our expected frequencies, this tells us that the population is evolving. Okay, so just a summary. Hardy-Weinberg equilibrium describes a population that is not evolving, at least for the locus that we're talking about. And because our definition of evolution is change in allele frequencies, what we're observing is that allele frequencies haven't changed, don't change. Okay, alleles still sort themselves out in a random way. These are our assumptions. We encountered these in lab. We think if we meet all of these requirements, if a population rather meets all of these requirements, it's not evolving. We can sort of lump together no mutation and no gene flow in that we're not introducing any new alleles. Okay, so here's how they come in on our life cycle. As we transfer from gametes to zygotes, we're assuming that our population is large and that individuals are mating randomly. They're not mating with any bias towards the alleles that their potential mates have. As we go from genotype frequencies in one generation to genotype frequencies in the next generation, we're assuming that there's no gene flow, there are no alleles coming in or no alleles leaving. We're assuming there's no selection that allows some individuals to grow up and other individuals not to. As we go from individuals to gametes, we are assuming that there's no mutation, no allele. Big A's aren't turning into little a's, and little a's aren't turning into big A's. Or we're not getting any additional alleles. We're also here assuming no selection, that all individuals contribute equally to the gene pool. All right, so all of these assumptions need to be met for a population to be not evolving. Okay, so I've got 30 seconds. A review. We're given some information about the population. We calculate the actual allele frequencies. We know them. We've observed them. We then calculate the way we expect those allele frequencies to sort out into genotypes. And we compare that expectation to the actual genotype frequencies that we were given at the beginning. So we calculate, and then really important, we compare. All right. Assessment 3 is available on Course Connect. We've covered the whole topic now, so take this assessment and then we'll work through some problems together.